Mapping the immune system of a bird against a human reveals a strikingly divergent, highly specialized evolutionary path. Birds face a unique biological challenge. They must defend immense, highly exposed mucosal surface areas across their respiratory and gastrointestinal tracts, but they do so without the centralized, encapsulated lymph nodes that mammals rely on to filter pathogens. To compensate, they deploy primary lymphoid organs found nowhere else in nature and equip their first responder cells with unique pattern recognition receptors to constantly sense their microbial environment. Avian immunity is defined by a highly distributed defense network, a specialized mucosal strategy, and the distinct primary organs that build the adaptive response. We start at the dorsal cloacal wall with a primary lymphoid organ unique to birds, the Bursa of Fabricius. Inside this sac-like structure is a dense architecture of thousands of lymphoid follicles lining the bursal wall. This organ serves as the central manufacturing hub for the avian antibody-producing repertoire. It is the site where immature lymphocytes develop the ability to recognize specific pathogens. Because it sits right at a major mucosal gateway, this developmental process is protected by a specific host defense peptide, catalycidin B1, which guards the vulnerable cells taking shape inside. During embryonic development, multipotent stem cells travel through the bloodstream from the bone marrow to seed these internal follicles. Once situated, these precursor cells undergo rapid proliferation. Through a genetic mechanism called somatic gene conversion, these cells shuffle their DNA. This generates the diversity of surface receptors required to recognize millions of potential foreign antigens. After successfully maturing and proving their receptors work, these fully functional cells flow out of the bursa and into the peripheral circulatory vessels to populate the rest of the immune system. As the bird reaches sexual maturity post-hatch, the bursa undergoes programmed involution. The tissue naturally shrinks and is replaced by fat. Because these cells were first discovered in this specific avian organ long before the mammalian equivalent was identified, they are named B cells, with the B standing for bursa. The critical nature of this organ is why infectious bursal disease, or Gumboro disease, is so devastating. The virus targets the bursa directly, destroying the bird's ability to mount an immune response. This makes precise vaccination timing relative to early bursal development a priority in poultry medicine. The bursa has a temporary, defined lifespan. Once it has successfully seeded the secondary lymphoid tissues with mature B cells, its mandate is fulfilled. In mammals, lymph fluid is funneled through discrete encapsulated lymph nodes that trap and filter pathogens. Birds lack these distinct anatomical structures. Instead, birds rely on networks of mucosa-associated lymphoid tissue. This includes the Harderian gland in the eye, bronchus-associated tissue in the lungs, and dense clusters like the cecal tonsils in the gut. Rather than draining invaders away to a centralized screening location, this localized architecture intercepts pathogens exactly where they attempt to breach the body. Managing this diffuse network requires constant local surveillance. The tissues are loaded with pattern recognition receptors that continuously sample the local environment and the resident microbiota. This creates a delicate balancing act. The immune cells must maintain immune homeostasis, actively tolerating and nourishing beneficial gut bacteria, while remaining highly reactive to invading pathogens. By utilizing a decentralized, active mucosal firewall, birds handle infectious threats immediately at the border. The other arm of the adaptive immune system originates in the thymus. But rather than a single organ in the chest, the avian thymus is structured as a chain of 6 to 8 lobes running parallel to the jugular vein in the neck. Inside each thymic lobule, immature precursor cells arrive and undergo the rigorous process of maturing into T cells. This is a site of strict selection and central tolerance. Cells that fail to recognize targets or react to the bird's own tissues are eliminated, while successful T cells are cleared to exit into the bloodstream. Structurally, it looks different, but functionally, the avian thymus plays the same role as its mammalian counterpart, driving T cell maturation and establishing central tolerance. Once in circulation, mature avian CD4+ T cells differentiate into familiar subsets. They use homologous cytokines to form Th1, Th2, regulatory T cells, and Th17 cells, directing the broader immune response. Yet avian physiology maintains some distinct quirks. During a Th2 response, birds operate without functional eosinophils, and they do not produce IgE antibodies. The adaptive system ultimately relies on these two distinct origins. The bursa generates the B cell repertoire, and the thymus generates the T cell repertoire. Before the adaptive system engages, the innate immune system sends in its primary phagocyte, the heterophil, the avian equivalent to the mammalian neutrophil. These cells are the rapid first responders, migrating out of the bloodstream to actively locate, engulf, and destroy invading bacteria at the site of an infection. Under a microscope, they are easily identified by their characteristic rod or spindle-shaped granules packed inside the cell. Their biochemical toolkit operates on a different logic than mammals. Avian heterophils lack significant oxidative burst capabilities and do not contain myeloperoxidase. To kill pathogens, they rely on distinct non-oxidative mechanisms, deploying a dense arsenal of host defense peptides, specifically avian beta-defensins and cathelicidins, to physically disrupt bacterial cell membranes. They deploy these weapons based on a bird-specific sensor array designed to detect microbial viability. This array includes toll-like receptor 15, a pattern recognition receptor unique to chickens that activates in the presence of fungal and bacterial proteases. It also includes TLR21, the avian homolog used to recognize bacterial CpG DNA. When these receptors bind their targets, they drive signals down into the cell nucleus through the NF-kappa and MAPK pathways, triggering immediate defensive actions. The avian heterophil executes the defensive mandate of the mammalian neutrophil, but operates on an entirely different biochemical and sensory framework. Once an infection is cleared, memory B cells produce circulating antibodies. In birds, the primary systemic antibody is IgY, the evolutionary counterpart to mammalian IgG. This presents an intergenerational challenge. A newly hatched chick requires immediate immune protection before its own bursa and thymus have fully developed the capacity to make their own antibodies. The hen solves this through a transfer process. Circulating IgY antibodies are actively diverted from the bloodstream toward the reproductive tract. These defense proteins cross the follicular epithelial barrier, depositing in high concentrations into the developing yolk. Later, during embryogenesis, the stored IgY antibodies migrate from the yolk sac and flow straight into the embryonic bloodstream. By the time the chick is ready to hatch, it has absorbed a complete circulating library of the mother's immune experience. This targeted deposition of IgY bridges the developmental vulnerability gap, providing robust systemic passive immunity until the chick's decentralized network comes online. When we map the differences, the divergence is clear. Birds source their B cells from the Bursa of Fabricius, and they rely on IgY for systemic antibody defense and maternal transfer. Instead of encapsulated lymph nodes, they use diffuse mucosal lymphoid networks. On the front lines, the non-oxidative heterophil takes the place of the mammalian neutrophil. The avian immune system represents a highly optimized, independent evolutionary blueprint for host defense.